comes from Ms. Lewis from the University of Chicago after 7 years as the director of the Royal Botanic Gardens in Kew. It's actually a return to Chicago for him, where he spent almost 17 years at the Field Museum doing the work that made him most famous. And that's the title, and that's the topic actually of his seminar today, the origin and early diversification of angiosperms, that he's asking us where next.
I'm also asking you to overlook the typo in the title.
Yeah. Origin.
Yeah, so thank you very much for the invitation to be here, and particularly to those of you that attended yesterday, thank you for showing up 2 days in a row. You have great stamina. What I'm going to talk about today is something quite different. I'm still focused on flowering plants and their diversity, but asking the question now, not what it is today and where it's going in the future, but really how it came to be. And I was talking earlier about the role of diversity studies, and I think it is fantastic that we kind of, in a broad way, know how all of these major groups of land plants now fit together. But angiosperms really stand out. They stand out because of their diversity. Whatever the right number is, it's a very big number compared to all of the other groups. And they stand out too because they appear on the scene relatively late. Most of these other groups, all of these groups for which we have a fossil record, appear significantly earlier, at the beginning of the Mesozoic, whereas angiosperms, at least as we understand them right now, don't appear until the early Cretaceous. So they show up late, but boy, have they diversified since they've been here, and they've generated a huge variety of species. And of course, from an ecological point of view, they dominate the world's terrestrial ecosystems, and in terms of their interactions, maybe something like 1 in 3 of all of the species so far described on this planet, most of which, of course, are insects, feed on plants in one way or another, and because they're feeding on the dominant plants, they're feeding on angiosperms by and large. What I thought I would do today is to kind of summarize, from my perspective, where we've come from over the last few years. And back at the International Botanical Congress that was held in St. Louis in August 1999, I gave a plenary lecture and sort of summarized where we were with angiosperm phylogeny and so on at that point. And I thought what I'd do is sort of take that as a starting point and then kind of look and see where we've come since then. And I'll just reiterate quickly some of the points that I made in St. Louis, and then we'll kind of go forward and see what's happened over the last few years. In St. Louis, I really kind of emphasized 2 things. One, a lot of new and useful paleobotanical data, and we'll talk about that in a bit more detail in a moment. Second, macroevolutionary syntheses, that is putting angiosperms in a kind of broader context and seeing exactly how they did come to dominate the world's vegetation. And then third, the availability of increasingly robust phylogenies from the conjunction of 3 developments that have taken place in the course of my scientific career and the scientific career of many of us here. One, the kind of philosophical and methodological advances that led us to understand how we might reconstruct phylogeny. 2, the ease of availability of the computational methods necessary to do that. And third, the relatively easy and standard comparative basis provided by the possibilities of investigating comparatively the DNA of plants. In terms of paleobotanical data, the real breakthrough that Jim mentioned in his introduction yesterday was the emergence of a new source of information. Not only pollen grains, which we've known about for a long time and particularly exploited since the Second World War, and not only leaf fossils, for which we've known about much earlier than that, having been collected in the mid-19th century, but the possibility of extracting from somewhat uninspiring samples such as this actually floral material of angiosperms that is remarkably preserved. And the person who really kind of got this started was my colleague, Professor Elsa Marie Fries, and she did her thesis, as it turns out, on Miocene material from Denmark. And the technique had been, in Europe, the standard approach had been to sieve these sediments and get out the fruits and seeds. She had the opportunity to study some roughly Campanian-age sediments, let's say 80 million years ago, from Osen in southern Sweden. She applied the same techniques that she applied in the Tertiary, and out came an extraordinary assemblage of beautifully preserved fossil flowers. This is one of the first to be described, Scandianthus costatus, and you can see from the preservation, you've got the calyx, you've got nectar is preserved, you've got the gynecium, you've got the remains of the corolla, and you've got the andrecium. And based on this material, we can reconstruct these flowers in great detail. They're very old, and having that because the systematics of living flowering plants is based to such a great extent on floral morphology, it allows us to fit in the flowers into our understanding of modern flowering plants. In addition, because we've got the stamens, it means that we can get the pollen in situ, and therefore when we find the pollen dispersed, we can make inferences about what its parent plants might have been, and also because of the flowers, we can make some inferences on what pollination biology and so on might have been like. So it really was a pretty fundamental breakthrough, and then pretty quickly we were off and running and saying, well, if they occur in southern Sweden, they've got to be in other places. So quickly we were looking all over the world trying to find these, and they've got to be in older rocks, so we went into older rocks, and that's the way this sort of went. And obviously the availability of scanning electron microscopy to study this material makes a big difference. And so here's an SEM stub with little clumps of pollen material, not flowers in this case, but this is much older. This is from a series of localities that we found in Portugal. The first place we went was to eastern North America, actually to the same sediments that Jim Doyle had worked on for his postdoc in the late 60s and early 70s. We went back to those sediments and we were able to get flowers out of some of those localities. And then we went to Portugal and spent a very happy summer driving around the clay pits of central Portugal looking for localities that would yield the kind of material that we were after, and we came up with actually quite a lot of material. And obviously studying this stuff with the SEM means that you can get great details on the pollen grains. This is a particular thing called that we call pennypolis. And we can also investigate these pollen grains in great detail and really take our study of these pollen grains to the next level. Obviously, there are limits to what one can do with light microscopy, scanning electron microscopy, and particularly if combined with transmission electron microscopy, helps you characterize these things in greater detail. And we had lots and lots of flowers, and after the Potomac group in eastern North America and Portugal, we then started looking in some younger sediments, and we had a series of localities from Georgia. We now have localities in Kazakhstan, in Japan, some been described from Antarctica, and so on. And so this material turns out to be quite widespread. One of the things is that these specimens are not so rare. If you sit on an outcrop, as we used to do, and split rocks all day, hoping to find flowers, you find a lot of leaves and not many flowers. When you take these bulk sediment samples and sieve them, you might end up with thousands of flowers. That's not to say that makes them necessarily easy to figure out. This is one locality from Georgia where we've been able to figure out some of these things. One's a conifer, two others are fagaceous things. These three, we don't really know what they are yet. And we have, as I'll mention at the end of the talk, we have lots of specimens where we only have one or two. And those we're starting to investigate in a bit more detail now too. But the point is that in some cases you can get a lot of this material, and also every bag is different. It's not very gratifying collecting, I might say. I mean, when we go to the localities, we're collecting pretty uninspiring stuff, and it goes into freezer bags, and then eventually, several months later, we would get it processed and find out what we've got. So we collect a lot of material in the field, and we don't really know what we've got until we get it back to the lab and process it. By experience, we know the kinds of sediments that we're looking for. And the time interval that we've really focused on is this kind of critical phase, during which, I mean, if you're looking at standard fossil floras of this age, you're not finding any angiosperms. When you get to fossil floras of this age, angiosperms are everywhere. And so it's really getting a handle on this transition has been a particular focus. Some of the syntheses that we put together back in the 80s, the kinds of syntheses that invertebrate paleontologists do, flawed in all kinds of ways, but they give you the basic picture. And that is that angiosperms come in with a bang. Some groups seem to decline. Cycadophytes particularly decline. Conifers seem to be relatively little affected. And there's a big change going on, too, in the pteridophyte floras. Also, during the 90s, this was paralleled by an improved understanding of angiosperm phylogeny and how angiosperm diversity should be put together, and we could start to integrate the material that we were getting from the flowers with the modern phylogenies. So what has happened really since St. Louis in 1999? Well, there's been a huge amount of effort on the phylogenetics of extant taxa. Many genes looked at, many more taxa looked at, more fine-grained and better resolved phylogenies. On the paleobotanical side, new material coming in from from localities that perhaps hadn't been studied in great detail at the time of the IBC, particularly our material from Portugal, but also interesting this material from China that I'll say a little bit about, coming from the same localities. You almost pick up every issue of Nature or Science, and there's another amazing vertebrate from the lower Cretaceous of China. Plant material does come out of those localities too. And also, interestingly, a little bit of information emerging on some floras at low paleolatitudes, particularly from the Crato Formation of Brazil, which gives us an insight into a totally different floristic province and enables us to test some predictions about the kinds of things that we thought might be there based on looking at the dispersed pollen. And then we've filled in with a lot of new localities, some of which I've already mentioned. On the phylogenetic side, I won't dwell on that, but all I'll say is that angiosperm phylogeny now has sort of pretty much settled down. And in broad terms, that's not to say there's not lots of work still to be done in figuring out details. Also, some work still to be done in figuring out some of these recalcitrant polycotomies, but there's a good deal of effort being focused on that, and that's the work that's being done largely by people interested in molecular phylogenetics. Those of you who've seen Nature over the last couple of weeks will have seen the story on Hydratella, which is a pretty rare example of a really major and surprising reorganization, where this rather unremarkable group of tiny little plants have been rather dramatically realigned from a position relatively close to grasses to a position just about at the base of the angiosperm phylogenetic tree. You'll hear a lot more. I think many of us just didn't have this family on our radar screen, even as much as as recently as 12 months ago. But you're going to hear a lot more about this peculiar family of two genera. And the reason is that it fits in here as the sort of sister group to water lilies. And this is a slide from the News and Views that Elsa Marie and I did in Nature in response to the paper by Jim and his colleagues a couple of weeks ago. And you know, it could hardly be more spectacular. I guess it would have been nice if it had been right at the base, but I mean, it's pretty close to the base here, and it doesn't look much like a water lily at first glance, but it appears to fit there robustly, and it introduces a new level of diversity into our understanding of the water lily clade. But by and large, those kinds of rearrangements now are becoming increasingly rare. And angiosperm phylogenetics is settling down, and pretty much we know where things go. We have lots and lots of flowers from the late Cretaceous, and figuring out what they are is actually not so easy because if they're down at the base of angiosperm phylogeny, the things that you have to compare them with are not too diverse. By the time you're getting up into the middle of angiosperm phylogeny, the diversity is huge, and figuring out exactly where these things go is certainly more challenging. So there's a lot of work still to be done on these late Cretaceous flowers, and I'll say more about that in a moment. Occasionally we get some interesting material. This is from the Chinese locality. It's very early. Little angiosperm carpels, obviously with seeds inside. 3 or 4 carpels fused at the base. But in a lot of ways, these fossils are kind of tantalizing, but we'd sure like to know what the pollen grains of this were like. We don't have that. And we'd like to know other kinds of details about it, which we often don't have. These are lake deposits into which plant material has drifted out, very nicely preserved at some level. In fact, in a lot of ways, better preserved than the material that we're dealing with, because often they give us whole plants, or at least pieces that are connected, whereas we always get little fragments. But our individual little fragments are much better preserved than these larger bits. The most famous of these is Archifructus, which found its way onto the front cover of Science, and highlights an issue that all of us have, and that I'll talk more about later, which is figuring out exactly how old are these things. And this was billed initially as Jurassic. I think we now understand that it's younger than that, and therefore doesn't stand out quite so much in the angiosperm picture as it would have done in the Jurassic. But this plant, Archifructus, has received a lot of interest, and we do actually know it pretty well. We know it from its leaves, we know it from its carpels, and we know it's from its peculiar stamens. And there's a lot of controversy about what it actually means, and indeed where it fits in angiosperm phylogeny. The initial idea is that it may fit below the point at which Ambarella and the rest of angiosperms diverged. Maybe it'll float up into and maybe floating is a good analogy for it. You see the fish in the background, may well be an aquatic. Maybe it'll move up into, inside the crown group of angiosperms. Maybe it'll even turn out to be close to Hydrotella. But figuring out how exactly it should be compared with modern angiosperms is one of the critical things in terms of looking at Archifructus, and indeed some other early flowering plants that we have. From Brazil, the material from the Crato Formation is preserved a bit like the material from China. Also, rather like China, a deposit that's collected and known mainly for its vertebrate remains, in this case fish. Same deal, plant material drifting out into lake deposits, often sort of whole plants with roots and leaves and so on attached, but often the preservation not really as good as one would like to pursue its relationships. Nevertheless, some interesting things there, and one of the things that we would have predicted would have been at low latitudes would have been lots of netales based on the dispersed pollen floras that we'd had from there, and they do indeed turn up in these deposits, and I think that's very, very clear now. So at low latitudes at that time, we're not looking at tropical rainforests. We're looking at a very, very different kind of an environment. We're looking at rather arid sorts of environments. Tropical rainforests come rather later, perhaps 70 million years or so ago. So in the context of angiosperms as a whole, this is a biome of relatively recent origin. Just some more material from the crater. So from Portugal, what are we getting? Well, we're getting things, the kinds of things from these early floras that you might hope to find. In other words, we're getting things that do appear to fit with the things that are down at the base of the angiosperm phylogeny based on molecular data. So we have flora material that we think is comparable to Nymphaeales, and we certainly have fruits and seeds of this kind with these characteristic exotestal seeds that are very, very similar to modern Nymphaealian seeds, which we can actually trace back from the recent through the Tertiary, through the late Cretaceous, and back into the early Cretaceous. So we know that the sort of water lily group was around. One of the problems that we have is with monocots. And one of the reasons for that is that they're not so easy to recognize. Their pollen grains can be very similar to the pollen grains of magnolias. But we do have a few cases, and this is one, where we've got very distinctive pollen that we think is similar to that of a relatively early diverging group of monocots, the Araceae. But in general, our understanding of monocots and their place in this early angiosperm radiation, based on paleontological evidence, is not so strong at the moment. What I think we have moved towards is a situation where we can now start to calibrate some of the nodes on this phylogenetic tree. And I think it's true to say that if you went back 20 years, we would have relatively few data points on this side of the line. And we've got quite a lot now, and we're going to get lots more. And so we have the opportunity to calibrate the nodes on this cladogram. And that's helpful because to the extent that we can do that, that then provides a basis for calibrating trees and extrapolations based on molecular rate calculations. And to do that, you need some good calibration points, and I think we can now begin to provide them. Now, what I'm going to do in the next few minutes is to look particularly at Eudicots, and which is the major and most diverse clade of angiosperms. But before I do that, I just want to emphasize as a kind of control the record that we have for one of these early diverging groups of angiosperms, the Chloranthasian. We don't know exactly how far back they go, but these are the earliest mesofossils that we can find. And they have a pretty continuous record through. And one of the remarkable things of looking at this flora material has been that we have been able to fill in quite a decent early record of Chloranthasiae. This was predicted to some extent by the early studies of fossil pollen grains, and people recognized quite early that some of these fossil pollen grains looked like Chloranthasiae. Again, a family that wasn't really on people's radar screens back in the 1960s. But it's been wonderful to be able to actually show that in addition to the dispersed pollen grains, we have flora material of Chloranthasiae. It's a group of just 4 modern genera, Chloranthus and Sarcandra, Heliosmum, and Ascarina. And in particular, we have early Cretaceous material, characteristic little triangular fruits, pollen grains, little staminate inflorescences that we compare in great detail with modern Chloranthasiae. So it's pretty clear. What I want to emphasize to you is that by and large this stuff is small. Look at the scale bar on that illustration. These are little inflorescences. So these are very, very, very tiny fossils. And some of that tininess is probably a result of shrinkage during preservation. Often this material is preserved as charcoal as a result of forest fire. You do some shrinkage, but nevertheless, this is very, very small. You don't get this much shrinkage. One of the characteristic features of these early floras is the flora remains are very, very, very small. In slightly later floras, we do have some relatively large angiosperm reproductive structures. It may be that if we got into the right floras of the right age, we might see some bigger ones, and indeed the Chinese material and some of the Brazilian material points to some slightly larger material, but there's no doubt that a lot of early angiosperm reproductive material is very, very tiny indeed. Okay, so I'm going to talk now about Eudicots. And rather than starting at the beginning and kind of tracing them up, I'm going to do it the reverse. I'm going to start in the early tertiary and sort of trace them back, because I think the patterns are quite interesting. Eudicot phylogeny here, this is the one of Dick Olmsted's and Walter Judd reviewed in AJB back in 2004, and I'm going to use that as the kind of framework for discussing their appearance of these clades in the fossil record. First of all, if you look at angiosperm diversity as a whole, Eudicots account for the bulk of it. The magnoliids, Sensulato, not hugely diverse. Monocots, perhaps about a fifth of total angiosperm diversity, but Eudicots account for the bulk of it, and they contain a huge variety of different forms. About half of those Eudicots are in the Asteroid clade, a good chunk of it in the Rosids, and then some of these other smaller groups. So that too is worth bearing in mind. And some very, very big groups, and indeed some very, very big families. Legumes, Rubiaceae, the whole Asteroid clade accounting for almost half of Eudicot species diversity. And the wonderful thing about Eudicots, at least as a group, is that they're not only strongly supported based on molecular data, but they also have a good morphological synapomorphy by which you can recognize them in the fossil record, and that is the occurrence of this triaperturate pollen grain, which probably represents some fundamental reorganization of microsporogenesis that I don't think we really understand, that changes the polarity of the developing pollen grains from having their apertures positioned on the distal surface to having them positioned equatorially equally disposed around the equator. It seems to be developmentally, it seems to be the same mechanism that somehow is repositioned as a result of some basic change in microsporogenesis. So those triaperturate grains, when you find them in the fossil record, are good indicators of the presence of Eudicots. And for that reason, the first appearance of Eudicots is pretty well pinned down based on dispersed pollen. So these are a couple of pollen grains from the mid-Cretaceous, show very clearly the triaperturate condition. So if we go back to the Oligocene, let's say about 40 million years ago, we go to a flora that's pretty well known from Oregon, from the Bridge Creek. We have pretty good representations, just about all of these Eudicot clades, including the Asterids. We've got Caprifoliaceae, we've got Bignoniaceae, we've got evidence of Rubiaceae. And in the Rosids, we've got a lot of Phagales, Betulaceae, Juglandaceae, Myricaceae, Roseales, Malvaceae, Sapindales, Myrtales, and so on. If we go back a little bit in time, we go back to the London clay flora now. This is kind of Middle Eocene, so we're now back at about 50 million years. We've still got a pretty good representation. London clay flora, by the way, is very, very diverse. And I haven't put in everything in here, but I put in the major groups that are recognized based on fruits and seeds from the London clay flora. And again, you've still got a pretty good representation, although not so much in the Asterids. The Icacinaceae are definitely there, no doubt about that. Ericales and Cornelis are there, but some of the other groups, Bignoniaceae, Caprifoliaceae, and so on, that you do see in the Bridge Creek flora, we don't see there. They seem to come a little bit later. So let's just take a look at this Asteroid group and just in a very simplistic way take a look at how they appear in the fossil record. Well, looking first at the sort of Lameids and Campanulids, as I sort of implied already, their record back in the late Cretaceous is pretty sparse. The deepest record is based on Icacinaceae, an interesting family that diverges relatively close to the base of the Lameids. Some possible Aureliaceae, some possible Aquifoliaceae. Largely Maastrichtian, but with some indication that the Icacinaceae may go back a little further. This group is not well represented back in the late Cretaceous, despite the fact that we have lots of material in the late Cretaceous. This is not a group that we find. It's negative evidence, but on the other hand, at the moment, it's a pattern that seems to be there, and it's a pattern that's different from other asterids. So if we look at the Ericales, loads of evidence of them going back, and they're one of the early diverging groups of asterids. Lots of evidence of Ericales based on floral material now, Maastrichtian, Santonian, Campanian, back into the top of the Cenomanian. So pretty much as you'd predict from the phylogenetic analyses, Ericales are really quite important. They're the group of the asterids that radiates most obviously during the late Cretaceous. And we can recognize them based on fruits and seeds and flowers, and that record's pretty solid. Another group that you sort of expect to be there are the Cornelis, because they're the sister group to the rest of the asterids, and indeed they are there. We've certainly got them in the Maastrichtian. We now have described them from those floras in Japan. We also have them from those floras in southern Sweden. So the Cornelis too, going back not into the earliest phases of angiosperm evolution, but moving through the late Cretaceous into the early part of the late Cretaceous. Cornelian fruits are often fairly easy to recognize. They have a characteristic form of valvate dehiscence, and this is the material from the Coniacean of Japan. Now let's switch and have a look at the Roseids. And it's quite interesting that some of the Roseate groups that are easy to recognize don't seem to have a record that goes back into the late Cretaceous. Appropriate for Davis, the Vitaceae are well represented in the early Cretaceous. They're one of the characteristic elements in, for example, the London clay flora. They're very easy to recognize. Have a look at a grape pip sometime. Very characteristic in form, and that's what Vitaceous seeds look like by and large. We clearly have them in the early Tertiary, but we don't have them in the rich floras that we have from the late Cretaceous. And the same is true of legumes. And as you saw from that earlier diagram, the legumes are a group that's incredibly important, very, very diverse, one of the largest of all angiosperm families, and we really don't have convincing evidence of them going back into the early part of into the later part of the Cretaceous. Some hints of them in the latest Maastrichtian based on dispersed pollen grains. By the time you get into the early Tertiary, then you're picking them up as macrophoss. Maybe a recognition problem with the legumes. Maybe not so easy to recognize in the kinds of floras, but so far we don't have them back in the late Cretaceous. But we do have other groups. One of the groups that we very obviously have of Roseids back in the late Cretaceous are the what we call the sort of Normopoles families. We've known for a long time that these kinds of triporate pollen grains were around in the late Cretaceous, starting to appear not in the earliest Cenomanian, but around the mid-Cenomanian. Very characteristic morphology, very characteristic paleogeographically too, particularly of Western Europe at that time, with some representation too in Eastern North America. And through Else-Marie Fries's work and the work of others, and a little bit of work done in my lab, we now know what the flowers of these Normopoles were like, and we have quite a variety of them now. Many of them bisexual, but not all of them. Andresianthus, just described a couple of years ago, with unisexual flowers, and these are clearly related to the modern assemblage of families that includes the Fagaceae and related groups, Betulaceae, Juglandaceae, Myricaceae, and so on. So very distinctive pollen grains. We don't have them in the earlier sediments. We do have them from here on, and we do have them in flowers. And through this interval too, while we can't relate many of these things to particular families, we certainly can allocate them to this major group, the Phagalian group. But we also have things that we can identify. This is a little staminate flower belonging to Phagaceae. And we have the copules, and we have the fruits, and we have the staminate flowers. We can put the whole thing together. Here another one, Antiqua copula. This is material from Georgia. So we have pretty clear evidence of Phagaceae, Nothophagaceae in the southern hemisphere, largely based on dispersed but very distinctive pollen grains. Fits with this scenario. In the northern hemisphere, Phagaceous remains mainly based on floral material. Another group that's well represented is the Saxophrigales in the form of Hammermelidaceae, and we have good records of Hammermelidaceae through the late Cretaceous. This is one worked up by my student Susanna Magallon. Alonia, very distinctive arrangement and morphology of the stamens. And again, with this group, Saxophrigales, Hammermelidaceae, Altingiaceae, a group that's really a kind of a late Cretaceous group. So we've kind of looked at asterids, we've looked at rosids, but now what about these groups that diverge at the base of Eudicots? Well, pretty much as you'd expect, they do go back further. Most obviously, and one of the first things that we ran into were the platanoids. And they have a very good record. This is outlined in blue is material from southern Sweden, from the Campanian. Outlined in red is some material from North Carolina, from the Neuse River locality of Santonian Campanian age. But we also have it going back much further. Here from Georgia, from the Santonian, and then several forms from the Potomac group. So there's clear evidence of Platinacea, and these things start to now get back into the top of the early Cretaceous, pretty much as you'd expect from the molecular phylogenies. And there are other things there too that you might expect from the molecular phylogenies. This is a plant which I still have huge affection for, that we described back in the early 1990s. This is a thing called that we called Spanomera, and we figured it out in a fair amount of detail. This is a little staminate flower with 5 tepals, 5 stamens. The stamens look like this. A couple of species, the pollen grains are very, very distinctive. The carpels look like this. The papillae on the carpels look like this, and the pollen grains are nestled in amongst the papillae of the carpel. So we've got this thing reconstructed in a good deal of detail. This is what its sort of floral diagram looks like of Spanomera maldenensis. This is earliest Cretaceous in age, from the Malden Mountain locality in Maryland towards the top of the Potomac group. So little staminate flowers, basically set up on a dimerous plan with little pistillate flowers also there too. It fits very well with the modern family Buxaceae. And going back still further in the material from Portugal, we see older representatives of what seems to be the same clade. Pollen grains again with this distinctive vermiform sculpture, little bicarpellate gynecia, staminate, separate staminate and pistillate flowers, and again pollen grains. You can see them here in situ on the stigmatic surfaces, and this is getting back now into the early Cretaceous. By the time we get back to localities like Torres Vedras, which is getting back into perhaps as far back as the Aptian or maybe even the Barremian, we've got quite a bit of Angiosperm diversity, but most of it has monocolpate or monocolpate derived pollen, Chloranthasiae, this possible monocot. Eudicots are extremely rare, at that level. From Torres Vedras, one of the localities in Portugal, we only have, although we've got quite a bit of diversity at the monosulcate grade, we only have one type of triapterate pollen. Also occurs in coprolites. So in these very early floras from Portugal, some dispute about exactly how old they are, but they're certainly back in the early Albian and maybe back into the Aptian and Barremian. Eudicots are very, very subservient in terms of their relation to the overall angiosperm flora. And if you go to England, which perhaps has the best controlled section, Norman Hughes recognized several phases of early angiosperm evolution. These are all exclusively at the monocolpate level. You don't get triapterate pollen grains, and then they're extremely rare until you get to what he called phases 4 and 5. And they're known just from a few dispersed grains. And he was looking hard for them. In our Portuguese floras, if you tot up how many entities have we got, I don't know what they mean, species or something, eudicots are very much subservient to the monosulcates. So in the early phases of angiosperm evolution, the sort of distribution of angiosperm diversity was very, very different to what it seems like today. So the overall pattern, we have some groups that are going way back, certainly into the Aptian, maybe into the Barremian, things like Chloranthaceae. The early diverging groups of eudicots, Platinacea, Buxales, also representing the earliest appearance of eudicots. Phagales, a good example of a Roseate group with a good late Cretaceous record, but some of those Roseate taxa not coming until the early Tertiary. Cornelis and Ericales well represented in the late Cretaceous, but the record of Lemmiids and Campanulids much more equivocal. So I just want to spend the last 5 or 10 minutes just running through kind of where we've come from and where we might kind of go. Well, I think obviously we've made huge progress on a range of different fronts. I mean, back in '99, we could say that in terms of figuring out angiosperm diversity and phylogeny, we'd already made quite a bit of progress. By then we were starting to get the fossils, and it was starting to give us a sort of complete picture. In terms of the timing, we pretty much knew when the earliest tricolpates were around. We had no clear evidence of angiosperm stem groups prior to that, and we knew that angiosperms were exceptional in their diversity. I think what we've learned since then is I think we've been impressed by the accumulating evidence for the presence of some of the groups that you'd expect in the very earliest phases of angiosperm evolution. The chloranthoids we now, I think, have pretty clear evidence. Other things like nymphaeales were clearly there. We're struggling with monocots, in large part because we find it hard to recognize them. They're not distinctive enough that we know when we've got them. Unlike the eudicots, when we've got triapterate pollen grains, we know what we've got. But we also know enough that by the early late Cretaceous boundary, we've got a whole variety of different magnoliids, not only Chloranthaceae, but things that are clearly Lauraceae, Calicanthaceae, Magnoliaceae, Winteraceae, and perhaps Piperales. When eudicots do first appear, they're relatively rare at their first occurrence, subservient to the monocolpates. Where we can pin down their systematic relationships, they seem to make sense: Platinaceae, Buxales, and so on. Core eudicots, which comprise the bulk of extant diversity, seem to be basically a late Cretaceous group, and many of the most diverse eudicot clades, legumes and lemmiids, seem to have undergone their major radiation during the Cenozoic. So still a huge amount of evolutionary interest in looking at early tertiary floras, not for necessarily working out the early phases of angiosperm evolution, but if you're interested in asterids, there's still a lot of work to be done there. But I think it is important that we continue to test and sort of expand the paradigm. It's all very kind of comfortable at the moment. The phylogeny looks good. What we're getting from the record seems to fit. That's fine, but we need to continue to push it and test it for some of those reasons listed up there. One of the things I think that we should be worried about, and I think we should worry about these things, is we should be worried about to what extent is extinction messing up our understanding of the phylogenetic patterns. We know from looking at the record that there's a lot of extinction going on down there. We have groups that it's not so easy to figure out precisely how they fit with modern taxa. And what's the impact of that extinction on our processes of phylogenetic reconstruction based on extant taxa? I would suggest that all the indications is that it's a big problem in figuring out seed plant phylogeny, where we've only got 5 groups and we know we've got a lot of extinction. I would suggest that it could be a problem of significance at the base of angiosperms, but perhaps less of a problem by the time you get up into the rosids and asterids higher up. In those cases, more of the relevant diversity is still extant. The issues with sampling are probably less. But that, I think, all remains to be looked at in a good deal more detail. I think we've still got a lot of work to do on integrating the neo and paleobotanical data. And you know, one of the unfortunate things is that as people get interested in molecules, people don't tend to study morphology. Well, maybe that's okay, but if you want to put the fossils in, you've got to have morphology. And if you don't have that morphology, it's going to be very difficult to calibrate these trees and plug in the fossils. So there is still a need for good comparative, that is to say outward-looking rather than inward-looking studies of morphology on living plants. And we've got in the early Cretaceous, the problem is, what do these monocolpate grains represent? Are they magnoliids? Are they monocots? That's one of our issues. Later in the Cretaceous, it's a different problem. We know we got a lot of eudicots, but eudicots by then are so diverse that figuring out exactly where they fit, say, in a group like the Ericales, is not so straightforward. What are some of the weaknesses, some of the things that sort of frustrate me? Cretaceous is an enormously interesting time from a geological point of view. It's currently very difficult to integrate in what's going on the land with what's going on in the sea, because dating these floras on the land is very, very difficult, and changes are happening very fast through that mid-Cretaceous interval. Much of our oil, by the way, comes from rocks laid down in marine rocks of mid-Cretaceous age. There's a lot of interest in the kinds of changes, both tectonically, in terms of ocean chemistry, in terms of climate, that are happening at that time. We'd love to know how this angiosperm radiation fits into that. The difficulty is figuring out the stratigraphy and integrating what's going on with land, what's going on in the ocean. So that's a problem. Also, I think we've sort of stalled on seed-plant relationships. I'm not convinced that we've really got this cracked at the moment. We've got lots of key groups of extinct plants that seem relevant, things like Catonia, possible Netalian relatives. We still haven't figured out what the closest seed plant relatives to angiosperms are, and until we do that, we should always be worried, I think, about what the impact of that might be on relationships among basal angiosperms. We might expect from other areas that sooner or later we'll be able to do what we can do in other parts of the record, which is figure out the order in which some of the characteristic features of angiosperms were occurred. In other words, to find some angiosperm stem groups. But we haven't so far managed to do that. As is often the case in paleobotany, in the history of paleobotany, I suggest that we already have found the relevant plants, we just don't understand them well enough to figure out where they belong. And it is true that we are still grasping and grappling with the idea of how actually do you compare the flower of Welwitschia, say, with the flower of an extinct Bennettitalian, or with the flower of an angiosperm. There are many different ways in which those homologies might fit together, and we don't know which of those is correct. And so we do need a lot more detailed comparative morphological work to understand how these things fit together. Is this, for example, the equivalent of the whole thing, or is it just equivalent to some bits of this Bennettitalian flower? We could do with some more mesofloras from elsewhere. It'd be really nice to have the kinds of quality of floras that we have from northern latitudes down in places like West Africa, Brazil, Southeast Asia. We know the flora from the palynological evidence is different there. We don't have the details to sort of pursue it so far. One thing that I will just finish on, I think, is that we need a lot more effort on some of the gymnosperm, potential gymnosperm relatives of angiosperms. Can we extend the kinds of techniques that we've used in the Cretaceous, can we extend it further back, and can we use it to get a better insight into what's going on in some of these groups? I think we can, and I think we're going to need a lot more effort in that area. One of the things that we've started to explore recently, and I'll just finish with this, is the chance to characterize some of these fossils in more detail using high-resolution X-ray tomography. And that, I think, can be helpful in figuring out the precise details of the anatomy of these things. In other words, we can section them without having to embed them and section them. We can characterize them in 3 dimensions, and we're just really starting that work. And this is particularly helpful where we only have one specimen, or only a couple of specimens, because we don't have to destroy it as we prepare. I'll just give you one example from a paper that we will be submitting very soon. The images aren't as great as I'd like, but here's a little laureaceous flower, sometime in the Albion. But here is a section through one of its tepals using this high-resolution X-ray tomography. So we're getting cell-by-cell detail, and this is not as good as some of the more recent results that we've been getting. What you can do, of course, at that is to start to slice the flowers and then reconstruct them in 3 dimensions. The green are the tepals, the yellows and the oranges are the andrecium, the blue is the gynecium. And you can particularly get sort of internal structures, like you can see the ovule inside the gynecium. And this is pretty good. You know, these are 110 million years old or something. And then you can rotate them, manipulate them, you can dissect them. So you can pull off the tepals and leave the andrecium there, and you can see the carpel. And you can also visualize what's going on inside the carpel, so you can kind of see the stylar canal inside. These are, again, sections through the gynecium at different levels using this tomographic technique. And I think that offers huge potential in terms of studying some of these very rare specimens. And it's a technique, there's just been one paper on it so far, published in Nature, looking at some animal remains, these early metazoan embryos. But we've been getting some superb results on these early flowering plants, and we look forward to extending that technology and pushing that technique back a little further, not only on the angiosperms, but also on some of the seed plants that go with them. So with that, I think I'll finish. I think we have reached a point where we can fill in a lot of details. The question is, can we make another breakthrough? And I think to make that breakthrough, we're going to need not necessarily new material, but we are going to need new ways of looking at the material that we already have, and new ways to think about how all this should fit together. Thank you very much.
So thank you, Peter. Any questions? Maybe people want to have to leave. They could run out. No?
When do you see the first zygomorphic flowers?
First sympatlous corollas are in the middle of the late Cretaceous. First clear evidence of zygomorphing, I'm quickly racking my... We certainly have it in the early Tertiary, definitely clear. We have, for example, pretty good papilionoid flowers in the Eocene, so that's clear enough. We don't, if my memory serves me correct, have direct evidence of zygomorphic flowers in the late Cretaceous yet. The question is, to what extent do you want to kind of infer the presence of zygomorphic corollas based on the relationships of taxa that you already have? So, for example, we have what are obviously zingiberalian seeds. Zingiberales have zygomorphic corollas. Are we prepared to make that extrapolation? If we are, then we've got them in the late Cretaceous. I think it's likely, frankly, even though we don't have direct evidence for it. Yeah.
What are some of the gory details of extracting the mesofossils from the sediments?
Well, it couldn't be easier. I'm ashamed to admit. The trick with it is very straightforward, and that is you've got to find the right sediments. And in other words, you don't want, if they're too hard, forget it. It's just not worth the effort. They have to be soft. They have to be basically like modern mud. And the easier you can break them down, the better it is, because the harder you have to work to break it down, then the more impact that's going to have on the material. And it probably means that they've already been severely impacted by the kind of diagenetic process. So our tendency is, if it's too much like hard work, we go to another locality, where because you never know what's going to be in each bag. And frankly, even at the same locality where we've been back to try and replicate a good bag from a locality, proved enormously difficult. So it's very kind of stochastic what actually comes out of that sediment sample. But they're broken down largely in water, with maybe a little bit of persuasion from detergent. They have to be cleaned then in hydrochloric and hydrofluoric acid to get rid of any adhering mineral. But then you're really left picking through what looks like the sort of remains of tea leaves. But the things for the tomography, the really informative specimens for the tomography are going to be the fusionized charcoalified ones. And getting that kind of preservation, I'd love that to be the preservation in the Middle Jurassic of Yorkshire, for example, which is also very easy to break down from the famous Caton Bay locality, but the preservation is totally different. And so you're not going to be able to use tomography on those particular specimens. You will be able to use SEM, which has never been applied to that character. That'll be fine, but you won't be able to go to the anatomical step. Yeah.
Have you applied any of these studies to sediments from the Givingness fossil cliffs area? And what is the progress in preserving some of these areas as World Heritage Sites?
In the case of Givingness, which is obviously very famous for its vertebrate material, but also a lot of plant material there. I mean, some of these, I'm not sure about whether these techniques have been applied at Givingness. They have been applied, this is, by the way, much older material, Carboniferous material. They have been applied to some of the Carboniferous material in the UK. They have produced some interesting material, like I think still the earliest probable conifer is known from charcoalified material from the Carboniferous of the UK that was prepared in pretty much this way, although using HF rather than water to break the sediments down. I mean, the whole question of conservation of fossil localities is a huge issue, and I'm not sure I could kind of really get into it here, but there are some World Heritage Sites. I don't know whether Givingness is one, but the south coast of England, the classic Purbeck ammonite localities of Lyme Regis and so on is designated as a World Heritage Site, and doubtless there are others. And there are some restrictions on how collecting can go at those localities.
I asked the question because it seems that to achieve status as a World Heritage Site for places such as the Givingness Fossil Cliffs area, it's very difficult.
Well,
and yet these areas really shouldn't be preserved.
I mean, I have a bit of experience with World Heritage Sites because Kew was made a World Heritage Site during my time, and I can tell you that it's a largely political process. The basic view is that there's too many World Heritage Sites in the north and not enough World Heritage Sites in the south. Most of the northern countries get to put forward one nomination each year if they choose to do that. So the first thing that Joggins has got to do if it wants to be a World Heritage Site is to fight off all the other places in Canada that want to be World Heritage Sites too.
What's sort of the availability of grant money to do this sort of thing?
We'll soon see. In the past, it's been pretty good because I think that its implications are quite broad. And I mean, there's a huge amount of interest now in calibrating molecular phylogenies, and this is exactly the kind of data that you need for calibrating those things. And those calibrations allow you to go on and look at some pretty interesting evolutionary ideas. So I think it's absolutely possible, should be possible to get grant support for this kind of work. It certainly has been in the past, and I would expect it to be the case in the future, providing it can be framed in terms of the clear outputs that are going to come, and rather than just a total fishing expedition. By the way, Surveys and Inventories has funded this kind of work too.
How important it is to get paleobotanical and fossil data from the tropical parts of the world to complete angiosperm tree? Do you think it's going to significantly change, or because?
I doubt that it'll significantly change it, but there's certainly lots of things at low latitudes that we're clearly not understanding from our work at high latitudes. There are certain distinctive pollen grains, for example, at low latitudes that are kind of enigmatic, and we'd love to find them in their reproductive structures so that we could figure out what they really are. And it is very clear that it's a different kind of biome at low latitudes, a rather arid biome, full of potential angiosperm relatives like Nitales, and we'd like to know a lot more about them. So, you know, it's a choice that some of us will have to make. Do we want to stay home and work on the material in the lab, which we know is there and that we can work up, or do we want to go on a more speculative field trip where we might spend a month in the field and come back with nothing? On the other hand, we also might come back with some pretty exciting results, and that's a difficult trade-off to make.
Is the fossils as good as?
There's more of a problem with weathering, and that's absolutely clear. But there's no reason why we shouldn't, if we get into the right sediments in the right places, why we shouldn't come back with very similar kind of material, in my view.